So today we're going to do the blood PowerPoint video lecture. And we're going to go through the functions of blood. We're going to go through the different components of blood. As you remember, blood is a connective tissue, so there are cells, there's a liquid matrix called plasma, and then there are actually fibers. So we're going to go through all of the different things. We're going to start with roles and then go through all the different types of cells, and then we'll kind of end with a little discussion of blood typing, and that will be our blood lecture. So it's chapter 19 in your Martini textbook, and let's see if I can get this to go. There we go. So in the chapter, we're going to talk about what does blood do, and then we're going to talk about the plasma, the cells, and then on the right-hand side, you'll see what happens when we break a blood vessel. So we'll talk a little bit about tissue repair and how the body would heal a broken blood vessel. And then different types of, when we go all the way to the right, it says, what can the study of blood tell us about a patient? So we'll do a little bit about blood typing. So the functions of blood, or blood in general, is a connective tissue. And this is a really great first slide because it gives you the breakdown of the blood. It kind of tells you how much you should have, what a hematocrit is, what the packed cell volume is. And it's a really, really great slide. So it's a fluid connective tissue made up of plasma and then the formed elements. The formed elements are going to be everything from the cells to the platelets. And we'll talk about those specifically. You'll notice on the far right that 99% of the blood is red blood cells, or 99% of the formed elements is blood cells. And the plasma itself is mostly water, right? It's about 55% liquid, meaning 55% plasma, about 45% platelets and cells. I'm not going to read this whole thing because we are going to go into it, but it's a great first slide. It's a great summary in your book that you can take a good look at. So let's start at the beginning. The functions of blood for transport, regulation, and protection. Now we're going to transport oxygen, nutrients, metabolic waste, hormones, right? All types of things. And we're going to take those items all around the body. Each cell type, each organ is going to require different things, but the blood is sort of the main superhighway through the vessels of transporting things. Then we have regulatory functions. The blood, being such a large liquid reservoir, has a regulatory function for maintaining body temp, maintaining acid-base balance or pH, maintaining fluid balance, right, in and out of the cells, in and out of each organ. And then lastly, blood has a protective function, right, preventing blood loss, preventing water loss, and also helping to prevent and protect us from infection. So we'll talk a little bit about that today. We will talk mostly about that when we get to the lymphatic and immune systems. We'll talk about the white blood cells a little bit, but today we'll talk about them sort of just to list them and give you an idea about them. So the plasma portion of the blood, right? This is the liquid portion. 55% of the blood is plasma. Plasma is mostly water. And then all of these various types of solutes or dissolved substances, from electrolytes, and an electrolyte is basically a salt, sodium, potassium, calcium, magnesium, etc. Then we have plasma proteins, albumin, globulins, fibrinogen, different proteins that have different functions inside the body and inside the blood. We also have nitrogenous substances, and those are going to be byproducts or waste products, things that the body is trying to get rid of. Various organic nutrients, you can see there are a giant list of them, from amino acids to fatty acids to vitamins. Respiratory gases, mostly oxygen and carbon dioxide, and then steroid hormones and thyroid hormones that are carried around the body for various metabolic reasons. So the plasma is pretty important stuff, has a lot of components and moving parts. Now, the formed elements are going to be the red blood cells, the white blood cells, and the platelets. Now, a platelet isn't a true cell, okay? It's just a sort of a section of cytoplasm that breaks off from a larger cell. In this image here on the top, you can see the red, they're called biconcave discs. Those are the red blood cells. On the bottom image, those pink circles are the red blood cells. And then the larger, on the bottom, the larger ones with the giant purple nuclei are the various types of white blood cells. Same thing at the top. Those fuzzy, yellowish looking things are the leukocytes. So red blood cells and white blood cells are cells. Platelets are cell fragments. These are all make up the formed elements. So a red blood cell is going to look like this. It sort of is what's called a biconcave disc. So this isn't a hole. It doesn't go through the cell, so it just squeezes in. They're about 7.5 micrometers in diameter. Here is a picture of them when they sort of travel through the capillaries. You can see how they stack on top of each other, right? So they don't travel this way. They travel this way, stacked on top of each other. But in the larger vessels like the arteries and veins, they will sort of spin around. Most cells survive in the blood only for a couple of days. Blood cells are replaced by the stem cells located in the red bone marrow. So the cells, the red blood cells that sort of scoot around the body were created in the bone marrow and then secreted or excreted into the blood cells, and we'll get to that in a little bit. So the erythrocytes, biconcave in discs, the predominant role that they play in the blood is to carry oxygen, and the way that they do that is to bind oxygen to a molecule called hemoglobin. Now this is hemoglobin here on the right and on the left, but here on the right, and you can see how there is 1, 2, 3, 4 chains. Hemoglobin has 4 protein segments that fold up and clump up to make the one functional hemoglobin, 2 alpha and 2 beta polypeptide chains. What you can also see in the hemoglobin are these red, what's called the heme group, and that's what you see sort of pulled out here on the right-hand side. Heme is at its core, it's iron, and then all these various rings of carbon. And the heme group's role is to attach and hold on to oxygen. This is a very, very, very strong oxygen holding element as part of hemoglobin. So 97% of the red blood cell is hemoglobin. Here's another picture of the hemoglobin molecule in sort of its ribbon form. We see the hemoglobin and then the heme portion on the right-hand side. So when we talk about, I guess I can leave it here. When we talk about erythrocytes, right, they're basically the role of the erythrocyte of the red blood cell is to transport oxygen to the tissues. It has some role in taking carbon dioxide back, but not nearly as much as it does in carrying oxygen. The normal range for hemoglobin in the blood is between 13 and 18 milligrams per deciliter or grams per milliliter. And the oxygen diffuses into the blood from the lungs. So you inhale, oxygen diffuses from the lungs into the blood vessels. It attaches onto the red blood cell's heme, and then that red blood cell gets pumped around the body, and then the oxygen again diffuses into a cell that has a lower amount of oxygen in it. There's oxygen in it. That's hard to say. So about 20% of the carbon dioxide is carried in the blood on hemoglobin. It's called carbaminohemoglobin, but for the most part, we focus on red blood cells and hemoglobin as carrying oxygen. So how are the red blood cells made? It's a process called hematopoiesis, and you can see that here, hematopoietic stem cell. So we have a stem cell, and then if it's going to, so a hematopoietic stem cell is forms all of the different types of blood cells. So when it says stem cell, that means that cell can become anything, a white blood cell or a red blood cell or a platelet producing cell. If it's going to commit to become an erythrocyte, that's when we move into that lighter green, that medium green here that says a pro-erythroblast. So we have erythro, which means it's sort of on its way to becoming an erythrocyte. And then there are 1, 2, 3, 4, 5, right? We are really sort of going down this developmental pathway of erythropoiesis. Now it begins, right, when this stem cell is transformed into a pro-erythroblast, and then we end up going through phase 1, through phase 2, and here we start to accumulate hemoglobin. We really start to create and pump out that hemoglobin. And when that happens, right, most all of the other organelles right here, we're ejecting the nucleus. So the final erythrocyte is anuclear, 97% hemoglobin, because it differentiates all the way down this pathway, gets rid of all its other organelles, and becomes just a oxygen carrying machine. So it's a really, really specific and differential process to create an erythrocyte. Now, we don't want to make blood all the time, right? We want to make blood only when we need it. So there's a hormone called erythropoietin, which is called EPO. And EPO is produced mostly by the kidneys, and it's triggered by a variety of things, right? The body can create red blood cells under a number of conditions, usually to a low oxygen. So loss of red blood cells, if you have a blood loss for some reason. Insufficient hemoglobin, say you have some sort of anemia, and your hemoglobin isn't working correct, you don't have enough iron, right? So there's some sort of loss of hemoglobin. Reduced availability of oxygen, say that you're at a very high altitude or something like that. And sometimes high levels of testosterone can increase the EPO, which then increases the red blood cell formation. So there's a variety of different things. It basically comes down to, if the tissues need more oxygen for some reason, it will tell the cells to produce EPO, the kidney cells to produce EPO, which will then tell the red blood cell formation, or the hematopoiesis or erythropoiesis, to happen. So it comes down to a lack of oxygen for a variety of different reasons. Blood cells, red blood cells have a lifespan of about 120 days, right? Because they have no nuclei, they have no organelles, they're just out there, and when they sort of degrade and die, and that's what happens. This is a sort of a homeostatic control mechanism in regards to the production of red blood cells. So the first stimulus is inadequate oxygen delivery. Doesn't matter why, right? Decreased hemoglobin, decreased red blood cells, high altitude, doesn't matter why, right? But the tissues are feeling a little something. The kidney releases EPO. EPO stimulates the red bone marrow. The red blood cells are produced, and the oxygen carrying capacity of the blood rises. Now, this does not happen overnight, right? This takes days to happen. So this is not something that you will be able to repair within, a day or two, right? This is a pretty lengthy process. This is another way that we can view it, right? I like to include different images just in case students find they prefer different images. So here, again, low oxygen, erythropoietin rises, the red bone marrow produces red blood cells, etc., etc., etc. Now, that's the top 1 through 4. The bottom, notice how it says aged and damaged red blood cells are engulfed by macrophages of the spleen. So yes, we know what happens when we need more, but what happens to them when the dead and dying blood cells need to be, gathered up or recycled or whatever. So the heme is split from the globin, right? So you can see that right here at the beginning. So the aging and damaged cells are engulfed by macrophages of the spleen. The first thing you do is you split it up. The heme goes in one place, the globin goes in another, and then the heme is then picked up by the liver, and we can do a bunch of different things with it. Right? You can put it, it can be bound to transferrin and released, right, to be used again. And then, the bilirubin is picked up by the liver, and then it's secreted. And so basically the hemoglobin is broken down and reused, or certain portions will be excreted if the body's not going to use it. Again, right, the hemoglobin that's not phagocytized breaks down, and the alpha and beta chains are eliminated. If it is broken down, the bilirubin is bound in the liver, and then that is sent down through the bile to the intestine and again absorbed into the bloodstream. So the breakdown and the pieces parts are all sort of sent either to be reused again or to be excreted or secreted. All right. The next types of cells in the body are going to be the leukocytes, or the white blood cells. Now, the white blood cells are a very, very small portion, less than 1% of the total blood volume are white blood cells. Now, white blood cells, the whole role of white blood cells is critical to defense against disease. This is where our immune function comes in. The protective function of the blood happens when we start to discuss the white blood cells. Now, you'll notice that there are a lot of neutrophils, and then lymphocytes, and then monocytes, eosinophils, and basophils. And we're going to go through what each one of them do for the body. So here we have the 5 different images. And for a lab, it's important for you to look at these images because it is the best way to determine what type of cell it is by looking at the nucleus. You'll notice that each nuclei looks different, right? So we're going to go through each one of them bit by bit. So the first one here, we have neutrophils. So neutrophils are 50 to 70% of all white blood cells. They're the most numerous. They're chemically attracted to sites of inflammation, right? So any sort of cut or injury, we're going to have some neutrophils. They're phagocytizers. They're bacteria slayers. So what they're going to do, they're going to engulf bacteria, pull it in, and digest them, right? They actually pierce the plasma membrane of the bacteria and then kill it. Now, the way you're going to know which one it is, it's purplish, and it has between 3 and 5 lobes of its nucleus. All right, so that's a neutrophil. The next guy over here, the eosinophil, they have a bilobe nucleus, and they look a little bit more pink, right, than their neighbors. Eosinophils, only 2 to 4%. They attack parasitic worms, which hopefully most people don't have too often. The other really interesting thing about the eosinophils is somehow they play a role in asthma and allergy. And the asthma and allergy piece is important because asthma and allergy is really an autoimmune disease, right? It's really the body overreacting to something. If you have asthma, your lungs are oversensitive to things and they tend to tighten up. If you have allergies, your body tends to overreact to a protein that doesn't necessarily need to overreact to, right? If it's a peanut, like why is a peanut so scary, right? But your body somehow thinks that it's super scary. So eosinophils are being studied extensively in that role because they do play a role in asthma and allergy. And why do they overreact? So that's kind of an interesting thing. Here we have the basophil. Now the basophil has a U-shaped nucleus, right? And it's the rarest and least numerous. And this guy is responsible for releasing histamine. And histamine is an inflammatory producer. I wanted to come back here because what I didn't mention that there are 2 different groups of lymphocytes or white blood cells, leukocytes. One are called the granulocytes, these 3, and then the agranulocytes are these 3. And the reason I mention that is because these 3 are the granulocytes and these 2 are the agranulocytes. And basically it's supposed to say that you see these little granules, these cytoplasmic granules, but if I'm being honest, this to me doesn't look that much different than this. But under a microscope, these 3 are supposed to have more visible cytoplasmic granules. Now, then we have the agranulocytes, these 2 guys, lymphocyte and monocyte. Lymphocytes are 25% of all white blood cells. They're found everywhere, in the nodes, in the spleen, in the lymph vessels, but rarely they're found in the blood. We're going to talk about B and T lymphocytes over and over again in the immune system chapter. I think it's 22. But the T lymphocytes directly attack virus and cancerous cells. B lymphocytes produce antibodies, right? And that's this guy. You can tell it's a lymphocyte because the nucleus takes up almost the entire cell. It's a lymphocyte. Then lastly, we have our monocytes, 3 to 8% of white blood cells. They are also phagocytotic, and they're going to activate the lymphocytes. So these guys actually work together. but the neutrophil and the monocyte are bacteria producers. This is a great little table, right? It tells you what the cell type is, gives you a little picture of it, a description, right? How many cells there are, lifespan, and what it does. So this is a great little guide to have kind of around, printed out. Keep it next to you when you're taking your quiz, right? But it's pretty super helpful. Now, here we have that hematopoietic stem cell at the top again, and we already sort of followed it this way. Under the influence of EPO, we're going to get a red blood cell. The rest of this table shows you how that same stem cell can be influenced to become one of the 5 white blood cells, and also something called a megakaryocyte, which produces platelets. Remember that hematopoietic stem cell is going to give rise to all 7 of the formed elements inside of a cell, right, under a variety of different influences, MCSF, GM-CSF, right, all kinds of different things. But that stem cell can give rise to any of these 7 resulting cells. CSF means, colony stimulating factors, right? And that's going to be how what's called leukopoiesis happens. We're going to differentiate this myeloid stem cell, the lymphoid stem cell, into the various cell types at the bottom. A lymphoid stem cell is going to give rise to a lymphocyte, and basically the progenitor cell, or the myeloid stem cell's sort of offspring, progenitor cell is going to give rise to the other 4 types of leukocytes. And last but not least, we have our little friends, the platelets. Now remember I told you that platelets were segments of cytoplasm. They come off of a cell called a megakaryocyte, and they're not complete cells. They have a very short lifespan of about 10 days. Their role is for blood clotting, so if you get a cut, they will be these little fragments that kind of clump all together to prevent more blood loss. This developmental pathway is under the influence of something called thrombopoietin. A thrombus is a clot, so thrombopoietin is what sort of guides the developmental pathway of the megakaryocyte, which then can disperse its platelets into the blood. So they enter the blood, and the megakaryocyte sends the extensions through the wall called the sinusoid. When we get to vessels, we'll look at that. It's kind of interesting. It kind of just threads itself through the cells of the vessel, and then they break off and they float around in the blood. It's very kind of cool, actually. Now, the next thing was, so that was the red blood cells, the white blood cells, and the platelets. The thing about doing a lecture when I'm just sitting up here in my office/hallway is that I'm not writing it on the board. You're not asking me questions, right? It is sort of dry, and I do apologize for that. Maybe you find watching the videos helpful, maybe you do not. But I put them out there because everybody has a different mode of learning. So, backing up, we went over what is blood, the functions, the plasma, cell types. Now we're going to do a little bit about hemostasis, right? This is going to be how we prevent blood loss during a cut or a bruise. And basically, you'll see right here, there's a little cut. And the first thing that happens is called vascular spasm. And vascular spasm is the immediate constriction of the vessel injury, right? When the vessel knows it's cut, vascular spasm. After that, we have something called the platelet plug. And so here you'll see the platelets are starting to gather. And the thing about platelets, they release chemicals that make nearby platelets sticky. So one platelet says, hey, I need everybody, and starts gathering and sticking to all the other platelets. So it's a positive feedback loop. You don't see that very often in anatomy. Most of the time we have a negative feedback loop, but this is a positive feedback loop in which one platelet leads to more platelets, right? Lastly, we have coagulation, is which we have a protein called fibrin is then going to sort of form a mesh, and it's going to trap the blood cells and trap the platelets and create literally a Band-Aid inside, right, with all that stuff. From there, the body, now that it's not losing fluid, now that it's not losing blood, can take a minute. And from there, we have clot retraction and something called fibrinolysis, meaning the body will start to heal those cells from underneath, and then the clot will systematically be worn down and worn away until the vessel is healed to its full potential. Right? So the vascular phase, which is when the spasm happens, right? The endothelial cells become sticky, the platelets adhere to one another. Right here comes the platelet phase, right? The platelets are going to kind of make that bridge between the two broken pieces. ADP, PDGF, calcium, clotting factors, all of those things are sort of allowing all the platelets to aggressively come and make a nice sort of plug there. The coagulation phase, pretty complicated, right? The common pathway in blue, then there's the intrinsic and extrinsic pathways, but basically we're just trying to make a nice tight seal so that the cells underneath can grow and replace. Once the fibrin has been formed, right, the platelets contract, the clot begins to undergo clot retraction, right, and then it's all plugged up. And from there, right, the damaged tissue edges get pulled together and we have healed the cut. All right, the last thing I want to discuss is blood typing, because blood typing is important. You're going to do a little bit of it in lab and you want to have an idea of what's going on. So when we look at blood cells, and we know with human blood typing that we have 4 human blood types, right? A, B, and O. Now, this has them listed A, B, B, A, and O. And you'll notice that the one that's more prevalent is going to be O, followed by A, followed by B, and followed by AB. Now, what does the blood typing mean? What it means is that if you have a letter A, B, or AB, that means that on the surface of your red blood cells, you have a surface antigen. Now, in this image, it shows the antigen A as a triangle, antigen B as a circle. And the if you have A, your body will have B antibodies. So if you have B, your body will have A antibodies. So it's really about presenting the appropriate antigen to the body without initiating an immune response. So if you have blood type B and somebody tries to give you blood type A, it's going to cause an immune response. So A can only receive blood from A and O, because blood type O means you don't have any antigen on the surface of your blood cell. And you can see that down here. There's no circles, no triangles. On blood type A, you only have the A triangles. On blood type B, you have the B circles. And on blood type AB, you have both. There's different antibodies based on what you have. AB wouldn't have any antibodies because it wouldn't have an antibody to something that it is, right? It's only going to have the antibody to what it is not. So this is a great table. This is the blood group. This tells you which antigens are on the outside of the red blood cell. This tells you which antibodies are present. This gives you a picture. This tells you what blood can be received by that person. All right, and then this tells you the frequency in the population. You can see how blood type O is the most popular, most prevalent, followed by type A, and then B, and then AB. We also have something called an RH factor, and an RH factor is a different set of antigens that are either present or absent. There's only one, so it's RH positive or RH negative. This is another way to look at it, right? This gives you type A has surface antigen A, right? This type B has surface antigen B, type AB has both, type O has neither. Now, what do we need to know about this? So when we are testing or doing blood testing, we're going to try to attach on to that antibody. Attach on to that antigen. So when you're looking at a blood typing activity, if it gets all clumpy, it means that that person has that blood type. So for example, type AB is clumpy with A and clumpy with B. Blood type O is not clumpy, very smooth blood sample with O, right? Because it doesn't grab on to any of those antigens, it doesn't have one. So take a look at this, make sure you understand it, go to the module and watch any videos you need to watch to understand blood typing. And I think that this image also gives you a really great picture, right? Clumping, no clumping, and then it clumps with RH, so it's A positive. This one's B positive. This is AB positive. This is O negative. There's no clumping. So again, look at both of these. Take a good look at them because you want to understand what you're doing before you go and try to do the blood typing lab. Again, feel free to go through the quiz questions if you want to have a little quiz for yourself. I'm not going to go through them, but these quiz questions are in the plain PowerPoint in the module. Have a great day.